I am 100% sure you have heard and actually know about COVID-19. Well, it's actually an illness caused by a virus that can spread from person to person. It is a new coronavirus that has spread throughout the world, and its symptoms can range from no symptoms to severe illness. You can get COVID-19 if you are close to a person who has it within 2 meters. This virus can also enter the body if an infected person coughs, sneezes, or talks. You might also be able to get it by touching an object or surface that has virus on it, then touching your mouth, eye, or nose. Everyone is at risk of getting COVID-19. In fact, all the adults and people having serious health problems are at higher risk for more severe illnesses. The best way for us now is to avoid being exposed to the virus and wear a mask when out in public. This deadly coronavirus has caused huge problems in terms of health and mental clarity. As of May 2020, it is confirmed about 4 million people around the world has been suffering from this virus and causing almost 300,000 deaths. Admit it or not, this coronavirus has caused anxiety for people because we cannot really go out as often. This is due to practicing social distancing that make people feel lonely. Sadly, there is no effective vaccine to protect us against COVID-19, and infected patients should seek medical care to help in order to relieving the symptoms as there is no cure of COVID-19 at the moment. Hence, the focus of my study will be to select the suitable drugs out of 3, which are bleomycin, mitramycin, and goserelin, that binding with the coronavirus proteins. How about we use the computer to predict the interaction of this viral protein of COVID-19 with the possible drugs? This gives me the idea of docking process. I prepared the possible drugs that can be downloaded from PubChem. Bimol is also necessary to convert the save file to be seen in Autodoc tools for docking process. I've given with 3 drugs which are bleomycin, mitramycin, and goserelin. Bleomycin and goserelin are the drugs used to treat prostate cancer, while mitramycin is an antibiotic. The protein of this virus was also prepared from our CSV database in protein database format. I've removed the unnecessary molecules that might disturb docking process using Autodoc tools. Then I save it into a separate folder for all of the 3 drugs. Easy, we can proceed to docking process using Autodoc Vina. The main target is to block the activity of the viral protein. The drug interactions and viral protein was conducted and recorded. The protein for my study is SARS-CoV-2 chymotrypsin-like protease. There is a value of docking where it is named as binding energy value that is in kilocalorie per mole. Drugs with lower energy value are those which have a higher potential for protein interaction. For my study, mitramycin have the lowest energy and is the most preferable drug to bind with SARS-CoV-2 chymotrypsin-like protease. This value was obtained from Autodoc Vina. This will be set as my preliminary result at the moment. This is the best part about my research. Docking process plays an important role in selecting suitable drugs to cure diseases as well. It is not something new, and I believe that we can live mask-free later on without coronavirus. Hence, I will also try to analyze the binding sites and the molecular interaction for my drug-protein interaction. It involves with the binding of ligand with the viral protein of COVID-19. So I really believe that the docking process can also be successful for coronavirus. With that, ladies and gentlemen, thank you for your kind attention.